My love for plants actually started when I think I started my BSc honours course, and the option was to choose between BSc Botany and BSc Zoology. So I thought between the two, I thought plant was something that I was always keen to know more of, and therefore I chose plants, and so I took a BSc Botany. You look at the tree, look at the way it's structured, see the kind of environment that it needs to grow. Just like you would acknowledge a friend when you meet him or her on the road, you just want to look at a tree and acknowledge. I come from a family of businessmen. And I'm one of these odd men out because I realized that long time back that I would not make a good businessman. And that was certainly not my interest. So I realized that I had a flair towards sciences, and I pursued that interest. I've completed almost a little over 30 years at this institute. I joined here almost way back in the late 80s. And I was a young PhD then, just straight out of the university. My entire work that I do, my religion is what I do. This is what keeps me going. So for me, it's a way of life. Coming to the lab is just a way of life. Morning, everyone.
Good
morning. What's up? What's happening today?
I'm setting up that PCR, sir. I'm trying to do that PCR. That's not working since past week, 1 week, I guess.
We have to get this solved very quickly, because if we don't
Yes,
finish
sir.
this, our NGS fellow will stop doing this work.
Yes, sir.
Yeah, when email will come saying
I'm
that,
trying some other conditions now, and probably this will work this time.
Okay. Research demands a lot of patience. Many a times, the results that you obtain from your research, from your work, is not what you expect. So you tend to feel bad. A life of a scientist, one of the major things that you need to do is, write papers, scientific papers where you communicate your results to peer-reviewed journals. In that process, you have a lot of heartbreaks because your papers are rejected, or they ask you to do a lot more experiments to ensure that what you're saying is authentic. When you read a published paper, you only see what's successful. What is not successful, very little is written about it. To run a lab itself is a lot of problems, because machines break down, you need new machines, you need to communicate your requirements with other people, collaborate with other institutes to be able to use the machine and the facility that they have and which you don't have, and you need to use it to get results that you're aiming to get. So life is not that honky-dory. There's a lot of pitfalls, but having said that, it's part and parcel of life and of a scientist's career, and we do a lot of that. Just like any other living organisms on this planet, we are born, we conduct ourselves to a maturity, and during this course of life to death, we undergo through a lot of stresses. Plants also similarly undergo several stresses. So stress is a kind of a restriction, and most crop plants have enemies. Now, enemies could be in the form of bacteria, it could be in the form of viruses, it could be in the form of insects, and enemies could also be the environment in which it is growing. So my focus has basically been plants and its major stress factors, in other words, insects. Why insects? Because you will notice that insects consume a lot of plant material, destroy plant material, decrease yield. So the aim of any plant biologist ultimately is to increase yield, find out mechanism by which you can decrease this loss of crop yield and thereby increase productivity, ensure that the human population is not short of food. So I'm studying that, and this study can only be done by understanding the interaction between the insect and the plant. The insect-plant interaction is actually a constant battle. The plant might have resistance over a period of time because the insects have a much faster life cycle. They are able to produce several generations by the time the plant is able to finish one life cycle. And because of this huge numbers that these insects come in, they'll eventually, every resistance that you have against an insect, it will eventually break it down. So another interest that most of breeders and agronomists and agriculturalists would have is to increase the durability of resistance against insects. So to be able to do that, you have to first understand how is it that the insect can break down resistance. We chose a method, and not only did we choose a method, we also chose a plant insect model system. So we chose rice, and it's very obvious that why one would choose rice, because it's one of the most immensely cultivated or hugely cultivated crop plant in the world, and very high percentage of the world population is dependent on rice. But having said that, rice is also attacked by several insect pests. One of the major insect pests that attacks rice is the rice gall midge. All rice plants cannot resist all biotypes. Biotypes are basically different races of insects, if you may. You cannot differentiate them. You can only differentiate it depending on a particular resistant rice variety, whether the particular biotype can grow on it or not. But one of the problems that we are facing in the field today is how do we identify these biotypes? There is a conventional method which requires putting these biotypes on a rice plant, but it's a very time-taking and laborious process. So we wanted to have a faster, a more reliable method to be able to do this. So what we decided was to sequence the entire mitogenome of the rice gall midge and see whether we can differentiate gall midge based on the sequences of the mitochondrial genome of the rice gall midge. So we initiated a program on that, and it's a very interesting study because after a lot of struggle we got that to work, and we had reached about 95% of the complete sequence, and the remaining 5% or so, we could not complete because the system by which we are doing this analysis and sequencing just refused to work.
Mrs. Bates.
I guess my love for movies started before my love for science. With the group of friends that I was moving around with, all of us were very keen watching film. And the particular genre of movies that I like are basically thrillers and murder mysteries.
They're probably watching me. Well, let them. Let them see what kind of a person I am. I'm not even going to swat that fly. I hope they are watching. They'll see. They'll see and they'll know. And they'll say, why, she wouldn't even harm a fly.
My childhood was spent in Calcutta, and Calcutta being what it was during the 70s, there was a lot of good movies to see. Bridge on the River Kwai, Bicycle Thief, Lawrence of Arabia, Murder by Death. My like for murder mysteries might be primarily stemming from the fact that usually in a murder mystery you have a preamble or an introduction where a murder or a series of murders take place, and then there are clues strewn about which the investigator then looks into, and finally at the end of the movie, using those clues, he solves the crime. when we were trying to sequence the entire mitogenome of the rice gallmage, and we had finished about 95% of the work, and there was a small portion to which we were just not getting the last bit. So it took us about 40 days, 45 days before we actually got the answer. And again, I allude to the murder mystery, where there is one clue that you have missed, and that is the clue that actually solves the puzzle. So it almost turned out like that, because I remember I was stuck at a traffic, and I was thinking of the issues where other mitogenomes from other insects had similar issues, where some parts were switched around and were not forming, going in a particular way that is commonly known to go. So we had that idea, which I knew we hadn't tested. So that night I couldn't sleep, because I was just so keen to run back to the lab and test this hypothesis, whether that could be the reason. And I remember the next morning I was eagerly waiting for the student to come to the lab, and I said, there's one more thing. So I still remember she looked at me and said, yet another idea. So I said, let's just give this last try, because we are stuck. We are at the most, this is also not going to work, but let's just give it a shot. So I explained the hypothesis as to why we are testing out this particular experiment. And she said, yeah, we hadn't thought of that, let's try it. So she went ahead and set up the experiment, and by 12 noon, we got to see that it did work. And there was a big scream from when the gel was run, and she looked at it from under the UV, and she said, sir, we have an amplification. So now we have a technique by which you can actually look at differences, or methods by which you can differentiate the different biotypes. To cut a long story short, I'd like to say it is morphologically impossible to make these differences between these different biotypes. The next question that anyone would ask is, why do you need to differentiate these biotypes? The answer to that is very simple, because the resistance gene that is found in rice cannot resist all the biotypes. Some biotypes it can resist, some biotypes it can't resist. So therefore it becomes important and imperative that wherever rice is grown, you want to know what is the population of or which type of biotype is prevalent there. So there's a direct use of this kind of work at the practical level, where you can tell the farmer, advise the farmer as to what crop he should grow. Another main feature that is happening in the environment today, insects are picking up a lot of insecticide resistance. So the insecticides that you're normally spraying to get rid of these insects are now building resistance against those. So no one understands the phenomenon. How is it that the insects are able to pick up this resistance or develop resistance against these pesticides in such a short time? So some of the research that we are doing probably will hint at or give results or give answers as to how these insects are able to resist insecticide. We also, in the process of discoveries, we help a lot of people who come to our institute to gain experience and be trained. So we do a lot of training. In fact, there are a lot of young people who come here for their PhDs, and after a few years of being with us, they get their PhDs and go. So this ability to train people to conduct research and be independent researchers subsequently as they grow in their career is not only a challenge, but it's a highly gratifying experience. RNA is in WBPH.
His pet dialogue is Banda Ban. Like, become a human being first and then grow.
He's very disciplined and organized.
He would always want his students to engage in other activities as well.
So he likes to do, everything in a manner.
Don't forget your morals, don't forget your ethics. Stay tuned to the ground.
You don't have to keep yourself engaged in research so much that you miss out on other activities, what's happening around.
In science, everything you have to note down, right? So whatever we are doing, experiment-wise, like daily, whatever we are doing, he knows everything and he notes down everything.
You should know what's happening outside the lab, be it sports, be it news, be it science, anything that's happening.
Photography is something that I do as a pastime. I basically shoot inanimate objects. I like monuments, old monuments, and I like plants, flowers, insects. These are some of the recent photographs that I have taken. If you look at this photograph, where you can see these reptiles, and I found it very interesting. It's a dynamic process, almost looks as if one of the reptile is saying hello to the other one. And then these pristine and lovely forests, you can see a lot of lichen growth on the trees, and you can show this that there's probably very little pollution in them. And this is another good photograph of a fallen tree trunk in the forest, and you can see I've been able to get good amount of depth in the photograph because it shows the crack from the south zone of the photograph right up to the north. This is from one of the forests near Slough. I can't recall the name of this place, but if you look at the tree trunks here, it's got a hollow. Not only that, it looks like a creature from one of the Peter Jackson movies, where you see these animals as if this tree is going to just get animated and start walking. Morning. Yeah, I've not finished yet. MK is just coming. He's gone next door. There is some ceremony. So he'll finish that thing and come in. We are going together. Yeah, so we leave at 6:30, sir.
I always mention that he is Buddha Bargach. Buddha Bargach is nothing but an old banyan tree present in the end of the village, and he has so much of experience. Lots of things has happened.
So we don't work for someone, we work as a science. We enjoy science like that, whether somebody is asking us or not. And if you ask about Suresh, the number of hours he puts is a maximum in the ICGEB.
And then whenever we have any challenges, he's the person who comes forward and try to solve the problem like it's his problem.
So when I am leaving, Suresh Nair is here. When I am coming here, Suresh Nair is here. So when he went home, I don't know the thing like that.
If someone asks me the question, why does science excite you so much? The first thing that comes to my mind is it's basically an extension of your thinking. This excitement where you see that the results are being unraveled right in front of your eyes, where you just thought that this could happen. Your hypothesis is actually working in terms of the science that you have planned. What more excitement can you get? And this excitement, I think I would have probably got in no other profession. So if you ask me the question, what does it feel like to be a researcher? I would simply say it feels like being on top of the world. I don't know if there is another life. If I was given another life, I'd probably choose the same thing. It's been a long innings here, and it's been a wonderful innings being at ICGEB. It's been a tremendous learning process. These 31 years that I've spent in this institute has just been exhilarating. Now that innings is coming to an end. And I often wonder what next. And once I superannuate, what next is the question that I often ask myself.